Have you ever been teased by a friend of how short you are or when your parents are so tall? When all of your siblings are blessed in height and you wonder why you're different? Or it could be the other way around. Well, to answer your friend, you just say it's heredity. Good day everyone, my name is Rabbi John R. Santos and I'm going to be your assigned lecturer for this course. Today, we'll be discussing about the overview of genetics, specifically the Mendelian patterns of inheritance. This lesson talks about how the world first understood the transmission of traits between generations and the underlying principles surrounding genetics. So, what are the learning outcomes of this lesson? At the end of this session, students are expected to know the brief history of Mendel's experiments, understand the concepts surrounding the 3 Mendelian laws of genetics, and perform monohybrid and dihybrid test cross problems. So, this just doesn't serve as the objectives of this lesson, but it also serves as your topic outline. Before we discuss Mendel and his three principles, it's best that we establish our understanding on genetics. Genetics is the field of study that involves researching a living organism's genes. It concerns with how genes for a trait are inherited and its variation. So when we say this, it means that we are studying the molecular structure and function of genes, gene behavior in the context of a cell or an organism, patterns of inheritance from parent to offspring, and gene distribution and change within population. We also mentioned that genetics is the study of how genes of a trait are inherited and how it varies. This because genetics involves studying heredity. Heredity is the process that involves the transmission of traits and biological information between generations of organisms. This basically means that heredity is the transmission or the passing of genetic information from parent to offspring. Now, what is the key difference between genetics and heredity? Well, to answer this, it mainly concerns on the word how. So if you are confused, note that genetics mainly concerns on how genes are passed, while heredity is just simply the passing of genetic information from parents to offspring. So we essentially study genetics because it helps us know the different genetic and hereditary diseases a person has. For instance, we use genetic screening, pedigrees, and karyotyping to detect if a person has a mutated gene. From this, genetics prompts us to develop drug therapies and other medications to control the effect of such diseases. And lastly, it improves the work of other societal sectors, such as in agriculture, where GMOs are made to improve the quality of crops. In genetics, it involves studying our DNA or our deoxyribonucleic acid. As you know, DNA is the biological blueprint of organisms because it contains our genetic information or genetic makeup. You can think of it as a molecule in your body that contains instructions on how are you going to look like. And from the word itself, it is a nucleic acid, which means these are molecules produced in the nucleus of a cell. You have other nucleic acids such as your RNA. But the difference between RNA and DNA is that RNA is single-stranded while DNA is double-stranded and helical in formation. That's why we call it a double helix in structure. Also, look at this image. If you were to extract a DNA, let's say from a strawberry, it will precipitate into a cloud-like substance when you perform a DNA extraction technique. But that's another topic to discuss. Going back, in our cells, a thread-like structure can also be formed. These are your chromosomes. More specifically, chromosomes are located in the nucleus of our cells, and chromosomes is where the many of sequence of DNA are located. What you need to know about this is that each chromosome is made of a protein and a single molecule of DNA. Meanwhile, the units of heredity are called genes, and these are a portion from our DNA molecule that codes for a protein. These are called units of heredity because it acts as a biochemical instruction to make molecules called proteins. Remember, nucleic acids not only function for our body's instructions, it can also function for protein synthesis or protein making. These proteins, in turn, control the characteristics that create much of our individuality. To help you visualize DNA, here is a diagram distinguishing them. First, we have your cell, and inside the cell, it contains the nucleus, this dark purple circle inside. Second, the nucleus contains the thread-like structures known as your chromosomes, which contain the many sequence of your DNA. DNA, your biological blueprint, contains your genetic information, and it's this long strand here. Now, a portion of your DNA is called a gene, which is the basic unit of heredity, and it's this bracket here. Just note that this GACT is the different nitrogenous base pairs found in the DNA. Adenine pairs with thymine, and guanine pairs with cytosine. Again, to simplify, the nucleus contains the chromosome. The chromosome contains the DNA, and your DNA contains your gene. Speaking of genes, an allele is the different or alternate versions of a gene. In Mendelian genetics, you'll hear this word a lot. For example, you have a gene that codes for eye color. One allele may code for brown eyes, while another allele for blue eyes. Another example would be hair color. One allele may code for brown hair, while another allele for blonde hair. Now, the thing with alleles is that it comes in pairs for a specific trait like eye color or hair color to be expressed. Later on, allele pairing and expression of traits will be discussed. Prior to Mendel, most breeders believe parents of contrasting appearance always produce an offspring of intermediate appearance. This means that if a tall person were to have a baby with a short one, the baby would be medium in height. This inference before Mendel was a result of having no knowledge of cells or chromosomes, the idea that the offspring have traits of both parents, and reappearance of traits attributed to genetic instability. But during the 1850s to 1860s, an Austrian monk named Gregor Mendel was one of the first people to study the basic principles of heredity and later deemed as the father of genetics. Mendel was inclined to scientific learning, so even entering at a monastery, he did not let his religious belief get in the way for scientific discovery. In his case, he was always fascinated by the patterns in the natural variation of plants. He experimented with thousands of pea plants or Pisum sativum and established the basic foundations of inheritance. He described the units of inheritance as elementen that pass traits from generation to generation. Mendel could not see them and had no knowledge of DNA or genes. But he did identify that inheritance is particulate, meaning it depends on the transfer of separate factors from parents to offspring. Now, here's a trivia. I want you to think, why did Mendel use pea plants in his experiments? What's special in pea plants that Mendel saw? I want you to pause the video and think for a second. Well, there are three simple reasons. First, pea plants are very easy to grow. They are easily reproducible because they develop quickly. If I'm not mistaken, their life cycle is only around 60 to 90 days. Second, the pollination of pea plants can be controlled. The flowers of pea plants contain both male and female parts called the stamen and stigma, and they usually self-pollinate. So, with this in mind, Mendel's experimentation involved a lot of cross-pollinating plants and subsequently self-pollinating them to determine the offspring of parental peas. And for the third and last reason, Mendel noticed that pea plants have easily identifiable traits, meaning each trait is distinguishable from the other. Speaking of traits, he studied 7 traits in his plants that appeared to breed true. These are the seed form, seed cotyledon, flower color, pod color, stem place, and stem size. Note that in some references, flower color is substituted with seed coat color. In this table, round seeds are dominant over wrinkled seeds, yellow seeds are dominant over green seeds, white flowers are dominant over violet flowers, and so forth and so on. In terms of the seed coat color, gray seeds are dominant over white seeds. These traits Mendel studied in his crossbreds follow a single trait inheritance, which means he chose varieties that differed in only one trait, such as tall plants versus short plants. He began with plants that had been cultivated over many generations to ensure they always bred through. This means that parents would produce an offspring that would carry the same express trait or exterior appearance. He set up all possible combinations with cross-pollination, tall with tall, short with short, and tall with short plants. The one where he crossed tall and short plants produced hybrids, which means an offspring has an inherited allele different from each parent. He also self-crossed these plants. Mendel noticed short plants crossed with another short plants were considered as true breeding, always producing the same express trait of shortness. But he was confused with tall plants. Some tall plants were true breeding, but other crossed with each other produced short plants. In the next slide, I'll be showing you what he meant. In this diagram, note that we'll be focusing more on the right side. The left side just shows what most readers believed during that time, which is that parents of contrasting appearance always produce an offspring of intermediate appearance. But Mendel's experiments were contradictory to this belief. Going back, when Mendel crossed the tall and the short plant with each other, which is considered the parental generation or P1, it produced all tall plants in the phylogeneration 1 or F1. Afterwards, the F1 generation self-pollinated or self-fertilized itself, which produced 3 tall plants and 1 short plant in the second phylogeneration or F2. Mendel tried this with other traits, when he crossbred true white flowers to violet ones. The results were the same in the F2 generation with 3 white flowers and 1 purple flower. From here, the first 2 of the 3 laws were formed, the law of dominance and the law of independent assortment. The first Mendelian law is the law of dominance, which states that if one organism is heterozygous, the dominant trait will mask the presence of the recessive trait. This basically means that one trait will conceal the presence of another trait for the same characteristics. In order to understand this law, I've colored some important terms that is essential for you to grasp and to understand the succeeding slides. So first, dominant versus recessive. Remember earlier in alleles when I said allele come in pairs before determining its express trait? Well, the dominant allele is represented by capital letters and you only need one copy of that trait in a given pair in order to be expressed. Meanwhile, the recessive allele is represented by small letters and you need 2 copies of that to be expressed. So in this example, the dominant trait of tallness is the capital T, while the recessive trait of smallness is the small t. In the first example, both dominant alleles are presented in the first pair, meaning tallness trait will be expressed. The next pair, we have different alleles, a capital and a small one. But the tall trait will be expressed because it only needs one copy, unlike the recessive trait. And on the last one, since only 2 lowercase or recessive alleles are present, then the expressed trait will be small plants. Hence, we can add to the definition that dominant traits always masks the recessive trait. Next, homozygous versus heterozygous. Organisms that are homozygous have either both copies of the same dominant allele or the same recessive allele, while heterozygous refers to one of the alleles are different. So again, in this example, the 2 capital T's is homozygous, specifically homozygous dominant, because it contains both the dominant allele. TT is also homozygous, but homozygous recessive, because it contains both the recessive allele. Meanwhile, this capital T and small t is just heterozygous because it contains 2 different alleles. The last terms you need to know are not in the color definition above. Sorry. These are the genotype and the phenotype. Genotypes describe the organism's alleles because these are the genes inherited from the parent to the offspring. Meanwhile, phenotype describes the outward expression of the allele or the one expressed. So for you to remember, know that genotype are the non-observed. Our eyes can't see them. while phenotypes are the observed, or as I've been coining it as expressed state, because our eyes can see them. So in this example, the genotype for the first is TT, so its phenotype or expressed trait will be tall plants. The second type, its genotype, is capital T and small t, so its expressed trait will also be tall. And on the last one, since both of its alleles are the small ones, its phenotype will be the short ones. So, thus knowing these terms clarifies why the first Mendel law states this way. And this is also why Mendel's experiments produced 3 tall plants while 1 recessive plant. Now, the second law is the law of segregation, which states that a parent organism gives just one allele for a gene for each gamete they produce. This means that an organism can have 2 copies of every gene, and that an offspring can get 1 copy of each gene from each parent. But what are these gametes that the law is saying? Well, gametes are termed as the sex cells of an organism. For males, it is the sperm cells. For females, these are the egg cells. In other references, gametes are sometimes referred to as your germ cells. This diagram here is the expanded version of the diagram found in slide 10, and it also depicts how the law of segregation works. Remember, plants have male and female parts. Well, let's label the blue for the male gametes and red for the female gametes. The first step involves replication, where basically these alleles is dark blue and light blue T makes another copy of itself, giving 2 dark blue T's and 2 light blue T's. The same goes for the other one. They look identical, but they're actually homologous to each other. They are called sister chromatids TT, TT, TT. Thus, during meiosis 1, these are pulled apart from each other, or you can imagine you're splitting them apart to give rise to TT and TT. After meiosis 2 takes place and further splits this TT and TT, it will go to a single T, 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 and T. The same process goes for otherwise. Afterwards, when fertilization takes place or crossbreeding occurs, 2 gametes fuse together to create 2 copies of each gene to complete their genome in the F1 generation. So this T gets paired with this T gets paired with this T, and this T gets paired with this T, and this T gets paired with this T. So this is why the law of segregation states that a parent organism gives just one allele for each gene for each gamete they produce. Which of the parent's 2 alleles given to an offspring is random. In continuation of his experiments, Mendel self-crossed or self-fertilized the F1 generation tall plants. Again, with the law of segregation, replication makes another copy of the capital and small t gene to produce double capital and small t. And in meiosis 1, this is split apart. And in meiosis 2, it is further split apart to produce one allele or gamete of capital T, small t, and t. And because of self-fertilization, self-crossing, self-pollination, whatever you want to call it, it produces 3 tall plants that are homozygous dominant, 2 heterozygous, and 1 short plant TT during the F2 generation. Now, though it was amazing that Mendel was able to know how traits are inherited through the law of segregation, it wasn't exactly correct. Because the world, including Mendel, did not know the existence of DNA, genes, or how meiosis work, so it wasn't entirely accurate. We know this to be true because in future topics of this course, in the non-Mendelian inheritance part, we see how other events deviate the Mendelian laws he discovered. So the process that we discussed on the law of segregation involves performing a monohybrid test cross. A monohybrid test cross is the process by which you cross 2 organisms for a mono or single trait. So we have a sample problem here. In pea plants, the dominant seed color is yellow, while green is the recessive. Mendel crossed a homozygous yellow pea plant to a heterozygous yellow pea plant. What is the genotypic ratio, phenotype, and phenotypic ratio of Mendel's cross? So our first step is to identify which is the dominant and recessive trait. So according to our problem, the dominant trait is the yellow color, and the recessive trait is the green color. We indicate the dominant with a capital Y, and we indicate the recessive trait with a small y. The second step involves identifying the genotype of the parents. The genotype of parent 1 is a homozygous yellow, which we know it will have two capital Ys. Meanwhile, parent 2 is said to be a heterozygous yellow, meaning we know that its genotype would consist of a big Y and a small y. The third step is to perform a Punnett square, which is just a squared diagram that is used to predict the genotypes of a particular cross or breeding experiment. Put the individual alleles of the father on the left column and the individual alleles of the mother in the top row. You can do this either way, it just depends on your preference, but I like it this way. Now, do the crossing like how would you do a FOIL method in algebra class. So it goes like big Y times big Y, you'll get big Y, big Y. Big Y times big Y, you'll get another big Y, big Y. Small y times big Y, you'll get a big Y, small y. Small y times big Y, you'll get a big Y and small y. Next is to identify all the genotypes, which is 2 big Ys and 2 small y's. Next, we identify the genotypic ratio by counting how many it is in the Punnett square. You have 2 homozygous yellow and 2 heterozygous ones, which we can term it as 50-50 and simplify it as 1:1. Next, determine the possible phenotypes. In this case, it would be yellow. And since we know all of it would be yellow, our phenotypic ratio would be 100% yellow. Here's another problem you can work on. I want you to pause the video and try this problem for yourself before heading to the next slide. So, if you have performed the problem correctly, then you should arrive to these answers. Your genotype should be homozygous green and heterozygous. Your genotypic ratio should be 1 homozygous green and 1 heterozygous. Your phenotypes should be yellow and green, and your phenotypic ratio should be 1 yellow and 1 green. Here's another one. See if you can solve it. Pause the video again and try this problem for yourself before heading to the next slide. So, if you've solved the problem correctly, you should arrive to these answers. Your genotypes should be homozygous white, heterozygous, and homozygous purple. Meanwhile, your genotypic ratio should be 1 homozygous white, 2 heterozygous, and 1 homozygous purple. Meanwhile, your phenotypes should be white and purple. Your phenotypic ratio should be 3 white flowers and 1 purple flower. The third Mendelian law is the law of independent assortment. It states that the alleles of 2 or more different genes get sorted into gametes independently of one another. In other words, the allele a gamete receives for one gene does not influence the allele received for another gene. Let's look at an example with just 2 genes. Let's say that this parent cell is heterozygous for gene A and gene B, and this would be their genotype. When making gametes, the parent can only give one of the gene A alleles and one gene B alleles to the offspring. The crux of the law of independent assortment is which allele a gamete receives for gene A has no bearing on the allele it receives for gene B. So, if the gamete receives the dominant gene for gene A, the gamete is equally likely to receive the dominant or the recessive allele for gene B. These two gametes would be produced evenly in meiosis. Likewise, if the gamete receives the recessive alleles for gene A, it is equally likely to receive either alleles for gene B. So, these are all the possible gamete genotypes that could be created from this parent. And the law of independent assortment tells us that each combination has an equal probability of occurring because the allele you give a gamete for one gene has no bearing on the alleles that gamete receives for other genes. Furthering Mendel's experiments, he looked at the seed shape, which is either round or wrinkled, and seed color, which is either yellow or green. When he crossed true breeding plants that had round yellow seeds to true breeding plants that had wrinkled green seeds, all the F1 generation offspring produced round and yellow seeds. These offsprings were double heterozygotes or dihybrids of genotype RRYY. From their appearance, Mendel deduced that round is dominant to wrinkled and yellow to green. Next, he self-crossed, self-pollinated, or self-fertilized the dihybrid plants in a dihybrid cross, so named because you follow the two genes and it has a 4x4 grid instead of the 2x2 in monohybrid crosses. Mendel found four types of seeds in the next generation. 315 were round yellow, 108 were round green, 101 were wrinkled yellow, and 32 wrinkled genes. These classes appeared in the ratio of 9:3:3:1. And from here, Mendel selected the true breeding seeds, this RRY and this RRY gene, from the F2 generation, and it also produced the same combinations with the same ratio as this one. Now that you know the law of independent assortment, let's perform a dihybrid test cross. Again, a dihybrid test cross is the process by which you cross two organisms for two traits. We have a sample problem here. In pea plants, the dominant seed color is yellow, while green is the recessive. In terms of seed form, the alleles for round peas mask the alleles for wrinkled peas. Mendel crossed a pea plant that is heterozygous for seed color and homozygous recessive for seed form, to a pea plant that is homozygous recessive for seed color and heterozygous for seed form. Determine the offspring's possible genotypes and phenotypes and their respective ratios. To solve this problem, first identify which is the dominant and recessive trait for both genes. In this case, the dominant seed color is yellow, while green is the recessive. In terms of seed form, the alleles for round peas are the dominant traits, while the alleles for wrinkled peas are the recessive ones. Second, identify the genotype of the parents. The problem states that the first parent is heterozygous for seed color, so we know that it will have two different Y genes, and homozygous recessive for seed form, meaning it will have two lowercase genes. Next, the parent 2, according to the problem, is homozygous recessive for seed color, which will have two lowercase genes. Meanwhile, it is heterozygous for seed form, which will have two different R genes. Third, cross each genotype from each parent using the FOIL method. So you have something like this. Y times R, you'll have this. Y times R, you'll have this. Small y times small r, you'll have this and this. And so forth and so on. For the gamete combination of the father, you have YR, 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 YR. And for the mother, you have YR, 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 and YR. Place the father's gamete combination on the left side and the mother on the top. You can do this vice versa depending on what suits your taste. Now, cross or combine these gametes to each other. So, big Y cross to small y, you'll have this combination. Small r to big R, you'll have this. Big Y to small y, you'll have this. Small r to big R, you'll have this. Big Y to small y, you'll have this. Small r to small r, you'll have this. Big Y to big Y, rather small y, you'll have this. Small r to small r, you'll have this. And you have to do the following operations on the subsequent boxes. At the end of your dihybrid cross, you should arrive at these following gene combinations. Now, the sixth step involves listing all the possible genotypes of this cross. You'll get heterozygous yellow and heterozygous round, heterozygous yellow and homozygous recessive wrinkled, homozygous recessive green and heterozygous round, homozygous recessive green, and homozygous recessive wrinkled. Afterwards, find the genotypic ratio of this cross, which in the percentage form would be 4 or 25%. So 4 is equal to saying 25%. 25% heterozygous yellow and heterozygous round, 25% heterozygous yellow and homozygous recessive wrinkled, 25% homozygous recessive green and heterozygous round, and 25% homozygous recessive green and homozygous recessive wrinkled. We get these four on this. You just have to find it. Now you have to determine the possible phenotypes from your genotypes. At the end, you'll have yellow and round, yellow and wrinkled, green and round, and green and wrinkled. Now determine the possible phenotypic ratio of this. So from this, you'll have 4 yellow and round, 4 yellow and wrinkled, 4 green and round, and 4 green and wrinkled. Now, if we simplify this, you'll get the 1:1:1:1 ratio. Note that not all the genotypic ratio and phenotypic ratio is not always the same. It may differ. It's just in this particular problem, why is it the same? Here's another problem you can work on. I want you to pause the video and try this problem for yourself before heading to the next slide. If you performed it correctly, you'll arrive something like this. These are the possible gene combinations from parent 1, and these are the possible gene combinations for parent 2. Afterwards, your dihybrid cross should look something like this. Meanwhile, there's only one genotype, which is the heterozygous yellow and heterozygous round. So, its genotypic ratio would be 100% heterozygous yellow and heterozygous round. Then, obviously, its phenotypes will only be yellow and round peas. And it has a phenotypic ratio of 100% yellow and round peas. Here's another one. See if you can solve it. Pause the video again and try this problem for yourself before heading towards the next slide. If you follow the same process as earlier and arrive to 1:16 or 6.25%, then your answer is wrong. Well, technically it is right, it would be 1/16 or 6.25%. But analyze the problem question carefully. It says a heterozygous man for both traits is crossed with a sterile woman who is also heterozygous for both traits. A sterile person is equally saying that they are infertile. So, the correct answer would be 0% because sterile women can't be impregnated or conceive a child. Gotcha! If you're one of those who fell for this trick question, I apologize right now. But next time, be careful with this type of problems, alright? So this is the end of our lesson. Here's a quick summary point of our discussion. Genetics is the study of how genes are passed. Meanwhile, heredity is simply the passing of characteristics that are transmitted from parents to their offspring. The key difference between these two terms is simply on the word how. Next, through planting trees or Pisum sativum, don't forget that, Gregory Mendel was the first to study how genes are inherited. Because of his studies, the three laws of Mendelian inheritance was formed. Mendel's work was pivotal in understanding the human genome, as we apply in our everyday lives right now. The three laws of Mendelian inheritance are: 1. The law of dominance. It states that if one organism is heterozygous, the dominant trait will mask the presence of the recessive trait. Meanwhile, the law of segregation, a parent organism gives just one allele for a gene for each gamete they produce. And on the law of independent assortment, the alleles of two or more different genes get sorted into gametes independently of one another. To test your knowledge from this topic, here's your homework to be submitted a week from now in Canvas. It can be handwritten on any paper, or you can put your answers in a document file in MS Word. Here are the references for this presentation, and if you have further questions, clarifications on the topic, please don't hesitate to message me on our Canvas account. That's all. Thank you very much.